Today we are going to discuss about the transcription. What the transcription is. Transcription simply means RNA synthesis. Transcription is simply DNA dependent. Transcription is simply DNA dependent RNA synthesis. It is DNA dependent RNA synthesis. This is the concise definition of what the transcription is. Transcription means DNA dependent RNA synthesis. See how it is dependent on DNA. So we have got this. We know that genetic information always flows from DNA to RNA and then RNA to proteins. So this is the flow of genetic information and living organisms. So we have got this DNA. It can synthesize its own carbon copies. We call this replication. When DNA synthesizes its own carbon copies, we call this replication. And when RNA is synthesized, from DNA, we call this transcription. This is transcription. And RNA, which are synthesized from DNA, mainly are of three types, that is mRNA, it can be rRNA, and transfer RNA. Then proteins are synthesized under the instructions of RNA. We call this translation. So translation. So we see that this synthesis of RNA is called as transcription. And the synthesis of RNA, it takes place, it is dependent on DNA. It takes place from DNA. We call this, it is a DNA dependent RNA synthesis. So when during this RNA synthesis, that is during transcription, also base pairing rule occurs here. That means adenine pairs with Watson and base pairing rule is followed here. Adenine pairs with uracil and uracil pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. So here thymine is absent, and instead of thymine we have got uracil. So thymine, it is present in thymine in DNA, that has been replaced by uracil in RNA. So the main enzyme, as we have seen in case of DNA replication, the main enzyme was DNA polymerases, while the main DNA synthesizing enzyme is in case of DNA replication. In case of a transcription, that is the synthesis of RNA, the main enzymes are RNA polymerases. RNA polymerases are the main enzymes for RNA synthesis in transcription, that is RNA polymerases. So RNA polymerases, these are the enzymes which are involved in the synthesis of RNA, that is, they are involved in transcription. Then we have this, in case of, if we'll see, in case of RNA polymerases, in case of prokaryotes and eukaryotes, in case of prokaryotes, this RNA polymerase, it is represented by alpha to beta, beta dash omega and sigma factor. This is in case of prokaryotes, this RNA polymerase is represented by alpha to beta, beta dash omega and sigma. In this sigma subunit, sigma subunit of this core enzyme, it helps in binding of RNA polymerase to promoter. It helps in binding of RNA polymerase to promoter. In case of prokaryotes, the RNA polymerase, it is represented by alpha to beta, beta dash omega, and sigma. This sigma factor, this sigma subunit, it helps in binding of RNA polymerase to promoter. Then this omega subunit, it stabilizes the core enzyme. It stabilizes the core enzyme. It stabilizes the core enzyme. And then we have another. We have this core enzyme. This core enzyme is alpha to beta dash omega. This is the core enzyme, and this is it contains the active site for the synthesis of RNA. That means this core enzyme, it has active site for RNA synthesis, It has active site for RNA synthesis. So in case of prokaryotes, RNA polymerase is represented by alpha to beta dash omega sigma. This sigma factor, it helps in loading the RNA polymerase to the promoter, and omega stabilizes the core enzyme. This core enzyme, it is represented by this alpha to beta dash omega. It is the active site which contains the RNA, it is the active site which is actively engaged in RNA synthesis. So in case of eukaryotes, this RNA polymerase, it is there are three types of RNA polymerase in eukaryotes. One is RNA polymerase first, RNA polymerase second, and RNA polymerase third. There are three types of RNA polymerase: RNA polymerase first, RNA polymerase second, and RNA polymerase third. This RNA polymerase first, it is involved in transcribing ribosomal RNA. That means there are three types of RNAs in eukaryotes. This RNA polymerase first, it is engaged in synthesizing ribosomal RNA. Then RNA polymerase second, it transcribes what? Messenger RNA. That means thus this RNA polymerase second, it transcribes all the structural genes. It transcribes the open reading frames in genomic DNA sequences. That means it transcribes this structural genes, this RNA polymerase second. So RNA polymerase second is the most important enzyme for transcribing the structural genes. That means it transcribes structural genes. It transcribes what? Structural gene. And then RNA polymerase third, it transcribes tRNA, transfer RNA. And there are three types of RNA polymerases in case of these eukaryotes: RNA polymerase first, RNA polymerase second, and RNA polymerase third. And in case of prokaryotes, we have got only one RNA polymerase, that is represented by alpha to beta dash, and omega. Now, this was the regarding the RNA polymerases, which is the main enzyme for RNA synthesis. So RNA polymerase are main enzyme on RNA, is the main enzyme for RNA synthesis. The main enzyme for RNA synthesis. And then we know that this is there are two strands of DNA. We have got this is obviously this is 5' end, 5 prime, 3' end, or 3 prime. We have 3' end, 3 prime, 5' end. It has, we know that this is a DNA molecule. It has got two strands. One strand is in the 5' to 3' direction, another strand is in the 3' to 5' direction. Now we know that this DNA is double-stranded molecule, whereas RNA is single-stranded molecule. This RNA is single stranded. This is RNA is single stranded. This is double stranded, and this is single stranded. So it means that from the double stranded DNA a single strand is formed. Now question arises that which acts as template, which strand acts as a template for RNA synthesis. RNA synthesis. Which strand acts as a template for RNA synthesis? Remember that, like DNA synthesis. DNA synthesis always takes place in the 5' to 3' direction. RNA synthesis also takes place in the 5' to 3' direction. So we have replication takes place in what direction? Replication takes place in 5' to 3' direction. Also transcription takes place in 5' to 3' direction. Also transcription takes place in 5' to 3' direction. Also transcription takes place in 5' to 3' direction. That means the strand of DNA which has polarity 3' to 5', it acts as a template for RNA synthesis. That means, for example, we have two strands. This is one strand, this is another strand. 5', 3', 3', 5'. Among these two strands, which strand act as a template for RNA synthesis? This strand will act as a template for the strand. We can write the strand which has polarity 3' to 5' direction. 3' to 5' direction will act as template for RNA synthesis. So we can say that we have got two strands. One is moving in the 5' to 3' direction, another is 3' to 5'. So this will act as a template. This is called as template strand. Template strand. Now why this the strand which has polarity 3' to 5' will act as a template for RNA synthesis? Because RNA synthesis always takes place in the transcription always takes in the 5' to 3' direction. That means the RNA synthesis will take in this direction. So which strand will act as a template for RNA synthesis? The strand which has polarity 3' to 5' direction will act as a template for RNA synthesis, because the RNA synthesis or transcription takes place in the 5' to 3' direction, as also replication takes place in the 5' to 3' direction. Therefore this strand will be called as 3' to 5' strand will be called as template strand, and another strand which does not take part in the RNA synthesis is called as non-template strand. It's called as non-template strand. Non-template strand. Non-template strand. So we have, this is non-template strand. Now question arises that this template strand, it is also called as template strand, is also called as non-coding strand, antisense strand. What it means? So I will write down here template strand, also known as template strand, also known as what? Template strand also known as non-coding strand. Non-coding strand or antisense strand. And this has polarity 5' to 3' direction. Then another strand, what is called as This template strand, it is called as non-coding strand or antisense strand. Template strand, it is called as non-coding strand, antisense strand, and it has polarity what? It has polarity 3' to 5', sorry, 3' to 5' direction. It has polarity 3' to 5' direction. This template strand, it is also known as non-coding strand, and it has polarity 3' to 5' direction. Now we have non-template strand. Non-template strand, it has polarity 5' to 3' direction. 5' to 3' direction, and it is known as non-template strand is called as coding strand. Called as coding strand or sense strand. It is called as coding strand or sense strand. What it means? The strand which has polarity 3' to 5', the strand which has polarity 3' to 5' will act as a template strand, and the strand which has polarity 5' to 3' will be called as non-template strand. This template strand is also called as template strand, is also called as non-coding strand. Template strand is also called as non-coding strand or antisense strand, while as non-template strand is called as coding strand or sense strand. What it means? What it means? It means that, for example, we have DNA. This is 5' to 3', and this is 3' to 5'. This is 3' to 5'. Now the question arises that these are two strands of DNA. This is 5' to 3' strand, and this strand runs in 3' to 5'. Which strand will act as a template for RNA synthesis? The strand which has polarity 3' to 5' will act as a template for RNA synthesis. So this strand will act as a template for RNA synthesis. Template strand. The template strand. The strand which has polarity 3' to 5', the strand which has polarity 3' to 5', which will act as a template strand, called as template strand. This another strand, it is called as non-template strand. Non-template strand, and this non-template strand has polarity 5' to 3'. Suppose we have base sequence. Suppose the base sequence in the DNA is like this. We have adenine, guanine, cytosine, thymine, adenine, adenine, cytosine, guanine, guanine, cytosine, adenine. Suppose this is the base sequence. This is the base sequence on the one strand of DNA. Obviously, the base sequence on the other strand of DNA can be easily predicted. So we have what? Adenine pairs with thymine, guanine pairs with cytosine, cytosine pairs with guanine, adenine pairs with thymine, thymine pairs with adenine, adenine pairs with thymine, adenine pairs with thymine, guanine pairs with cytosine, cytosine pairs with guanine, guanine pairs cytosine, guanine, adenine pairs with thymine. So we have a base sequence in both of the strands is now clear that this strand is having this base sequence, this is having complementary base sequence. Now I told you that the strand which has polarity 3' to 5' direction will act as a template strand because replication takes place in the 5' to 3' direction. So we have this strand will act as a direction of transcription. Direction of transcription is 5' to 3'. This strand which has polarity 3' to 5' will act as a template for the synthesis of RNA, and RNA synthesis takes place in the 5' to 3' direction. So it takes place in the 5' to 3' direction, and the sequence would be thymine pairs with adenine, cytosine pairs with guanine, guanine pairs with cytosine, adenine pairs with thymine is absent in RNA, thymine is absent in RNA, so instead of thymine we have uracil. So thymine pairs with adenine. So adenine pairs with thymine is absent because instead of thymine we have uracil, and then thymine pairs with adenine, thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine, then guanine pairs with cytosine, and cytosine pairs with guanine, and thymine pairs with adenine. So we see that this the newly synthesized mRNA, this is called as newly synthesized mRNA. This newly synthesized mRNA has base sequence similar to the base sequence of non-template strand. See, we have AA, GG, CC, AA, AA, CC, GG, GG, CC, AA, and that means all the base sequences in case of newly synthesized mRNA are identical or similar to the non-template strand, except there is uracil in RNA and there is thymine in DNA. So we say that non-template strand is the coding strand or the sense strand. Because why non-template strand is called as coding strand or sense strand? Because nitrogenous base sequence of newly synthesized base sequence of newly synthesized mRNA is identical. It is similar to the base sequence of non-template strand. So non-template strand is known as what? It is called as coding or sense strand. So non-template strand known as sense strand. Non-template strand known as sense strand or coding strand. Except there is an exception. One, in DNA thymine is present, and in RNA uracil will be present. So we can say that why this non-template strand is called as coding strand or sense strand. We have seen that the base sequence of newly synthesized mRNA, it is similar or it is identical to the non-template strand. So we have newly synthesized mRNA, this is A, G, G, C, C, except there is uracil in RNA and thymine is in DNA. Then we have A, C, 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 G, G, C, C, A. That means all the base sequences, all the bases are identical to the or similar to the non-template strand, which has polarity 5' to 3 direction. So the strand which has polarity 5' to 3 direction, which acts as a non-template strand, this non-template strand is called as coding strand or sense strand. Whereas template strand is known as non-coding strand. Template strand is known as non-coding strand or antisense strand, whereas template strand is known as non-coding strand or antisense strand. This is the difference between coding strand, non-coding strand, sense strand. That means this non-template strand is called a sense strand because of the fact that the nitrogenous base sequence, sense of mRNA is identical or similar to the base sequence sense of what? To non-template strand. So non-template strand is known as sense strand, whereas it is complementary sequence would be present on, it is the newly synthesized mRNA. It is base sequence, complementary base sequence would be present on template strand. So non-template strand is called as coding strand or sense strand. This is one point, that out of two strands of DNA, which strand will act as a template strand? The strand which has polarity 3' to 5' direction will act as a template strand, because transcription or RNA synthesis takes place in the 5' to 3' direction, and this template strand is also called as non-coding strand. It is also called as antisense strand, whereas this non-template strand is also known as coding strand. Non-template strand is also known as coding strand or sense strand. It has polarity 5' to 3' direction. And then this was which strand will act as a template strand, which is called as coding strand. Then we have got what is transcription unit. Transcription unit, it is a DNA segment that gets involved in the synthesis of RNA, that synthesizes RNA. What do you mean by transcription unit? What do you mean by transcription unit? Transcription unit. What is transcription unit? Transcription unit is, it is a DNA segment which synthesizes RNA. So transcription unit, it is defined as the segment of DNA which synthesizes RNA. That is called as transcription unit. How you can define a transcription? Transcription unit is defined as the segment of DNA which synthesizes RNA. That is called as transcription unit. This transcription unit has got three important components. One is called as what? One is called as promoter. One is called as promoter. Then another is called as structural gene. Structural gene. What is called as open reading frame. Structural gene. Then we have got terminator. This transcription unit is defined as the segment of DNA or DNA segment which synthesizes RNA. This transcription unit has three components. One is called as promoter, another is called as structural gene, what is called as open reading frame, and third is called as terminator. So we have transcription unit would be like this. Suppose we have this DNA. This is DNA, double-stranded DNA. This is 5' end. This is 3' end. This is 3' end. This is 5' end. Because of the antiparallel nature, we write two strands in opposite. They have opposite polarity. One strand is having 5' to 3' polarity. Another strand is having 3' to 5' polarity. This we write. This is the promoter. This is promoter. This is what is called as terminator. Terminator. This is promoter. We are discussing transcription. This is promoter. This is what is called as promoter. So we have, this is a DNA, double-stranded DNA. This is double-stranded DNA. And we have got what? This is promoter, this is terminator. This is called as, this site, this is called as transcription start site. This is transcription start site. This is transcription start site. And this site is called as, this site, it is called as termination, transcription termination site, transcription termination site. Transcription termination site. Transcription termination site. So this represents this region. It represents what? It represents structural gene or open reading frame. Structural gene or what is called as open reading frame. It is called as structural gene or open reading frame. This ORF represents open reading frame. Open reading frame. Open reading frame. This structural gene is also called as open reading frame. So we can say that this is the RNA polymerase. This is 5' to 3' end. This is, so this strand 5' to 3' strand. This is 5' to 3'. It is what strand? It is non-template strand. So this strand is called as non-template strand. Non-template strand. And the strand which has polarity 3' to 5', it will act as a template strand. It will act as a template strand. This is called as template strand because it has polarity 3' to 5'. This has polarity 3' to 5'. Now, this is transcription start site. Transcription start site. This transcription start site, this represents what? Upstream of the structural gene. This is called as upstream of the structural gene. Upstream of structural gene. And this is called, and this represents the downstream. This represents the downstream of structural gene. Downstream of structural gene. Now you can, how you can define the upstream of structural gene and downstream of structural gene? So upstream means that the direction, upstream direction means the direction in which the polymerase comes. That means polymerase will come from this side, from this end. This is called as upstream. And the polymerase will, that means upstream. How you can define this upstream? Upstream is the direction from which the RNA polymerase comes. That means it is a direction from which RNA polymerase comes. So we can say that this RNA polymerase comes from here. So this is the upstream of the structural gene. We have two strands. So we have this. We have two strands. In case of DNA, we have two strands. This is a non-template strand. What is called as coding strand, and another is template strand. Template strand. What is called as non-coding strand. So this RNA polymerase comes from 5' end. 5' end of the non-template strand. So 5' end of the non-template strand, and it comes from 3' end of the template strand. So we can say that this upstream for coding strand, then this upstream direction is 5', and for non-coding this is 3'. As you know that this RNA polymerase comes from this direction, so this becomes the upstream of the structural gene. Now we know that in the upstream we have got two strands, non-template strand and template strand. This is the 5' end of the non-template, so it becomes the upstream. We can say that upstream 5' is the upstream for the non-template strand, and this represents 3' represents the upstream for the template strand because of the it is because of the antiparallel nature of the two strands of the DNA. So same is the case. What is downstream? Downstream direction. Downstream direction represents downstream direction represents downstream direction. It represents the direction towards which the transcription proceeds. That means transcription proceeds. It is the direction towards which transcription proceeds. It is direction towards which transcription proceeds. So it means that this transcription proceeds towards this direction. Transcription proceeds towards this direction. So we can say that this 3 dash represents the 3 dash and represents the downstream for non-template strand, and 5 dash represents the downstream for the template strand. So this is what is called as upstream and downstream direction. This is this upstream and downstream, they represent the directions. That means the RNA polymerase comes from upstream direction and then proceeds towards the downstream direction. That means the direction in which the transcription proceeds, that is the downstream direction. And then this transcription start site, it is represented by this transcription start site. This transcription start site, it is represented by +1. It is represented by +1, and this transcription start site, it is represented by +1, and those which are towards the right side of the transcription site, we number them +2, and those nitrogen bases, those bases which are towards the left side, nitrogen bases towards right side, they are numbered as +2. For example, we have +2, +2, +3, +4, and bases which are towards the left, bases towards left side. That means this is transcription start site, and the bases which are towards the right side, this is towards the right side, we call them we write them +2, that is +2, +2, +3, +4, and the transcription start site is here, and the bases which are towards the left, we write them in negative. That means it would be like this, minus negative, that is -1, -2, -3. So it means that the first base which is transcribed, that would represent +1. The second base which is transcribed towards the right side, it is written as +2, and the third base which is transcribed, which is towards the right side of the transcription start site, it is represented as +3, and the base which remains untranslated and is towards the left side, it is written as -1, and the base which remains untranslated and is towards the left side, it is written as -2, -3. So negative sign represents the side which is towards the left side of the transcription start site, and positive represents side which is towards the right of the transcription start site, and transcription start site, we take it +1. This is what is called as upstream and downstream. Then we have got this, I said you that this promoter, I said you transcription unit. I told you that transcription unit has one is promoter, then we have got what? We have promoter, we have open reading frame, what is called a structural gene, and then we have terminator. This open reading frame, it represents a structural gene. It represents what is the promoter? Promoter is the binding site for RNA polymerase. It is the binding site for RNA polymerase. That means RNA polymerase binds to promoter, binding site for RNA polymerase. That means RNA polymerase, it binds to the promoter, and it is present, this promoter, it is present upstream of the structural gene. See, this is the structural gene or open reading frame. This promoter is present here, so we can say that it is present upstream of the structural gene. This is present upstream of structural gene. I told you that in case of prokaryotes, there is a factor that is called as sigma factor. sigma subunit. This sigma subunit helps in binding of RNA polymerase to the promoter. The sigma subunit in case of prokaryotes helps in binding RNA polymerase to the promoter. And in most cases, this promoter, it is having conserved sequence. It is having conserved sequence. And in case of a conserved sequence, that means, what do you mean by conserved sequence? This is the specific sequence which is conserved in the living organisms. That means it is constant, it is same in almost all the living organisms. For example, in case of prokaryotes, in case of prokaryotes, there is a that is called as pribnow box. This pribnow box, it is the sequence, it is the base sequence of 6. It is a base sequence of 6 nucleotides or 6 bases, I can say. That is TAAT 1, 2, 3, 4, 5, 6. It is the base sequence of this pribnow box. It is the base sequence of 6 nucleotides. This is TAAT. And this TAAT, this pribnow box, it is conserved in case of prokaryotes. It is present in the promoter region. It is also called as -10 base pair. It means that it is present 10 base pairs left. It is present 10 base pairs left to the transcription start site. So if this is a transcription start site, then we have to move towards the left side, and this left side, my negative sign represents the left side. These are 10 base pairs apart from the transcription start site. So it is highly conserved in case of prokaryotes. And this pribnow box, it was discovered by David Pribnow in case of prokaryotes. So this pribnow box, which is having the nucleotide sequence TATAAT, it is specific, it is conserved in the promoter of prokaryotes. And then we have got another in case of eukaryotes. In case of eukaryotes, there is also a conserved sequence in the promoter region, which is about 25 to 35 base pairs apart from the transcription start site, and that is called as Hoagnes box. This is called as Goldberg-Hoagnes box. Hoagnes box. This Goldberg-Hoagnes box, or simply Hoagnes box, it has a base sequence of TATA. It is also called as TATA box. So TATA box, known as Hoagnes box. This base sequence was discovered by David Hoagnes and his student, that is Goldberg, in case of promoter of eukaryotes. It is highly conserved, and it is about 25 to 35 base pairs from the transcription start site in case of promoter of eukaryotes, and this is known as Hoagnes box or Goldberg-Hoagnes box. So we can say that the pribnow box in case of prokaryotes and the Hoagnes box in case of eukaryotes, they are equivalent, and they are found in the promoter region. And basically these pribnow box in case of prokaryotes and Hoagnes box in case of eukaryotes, they serve as a site, as the base sequence, specific base sequences where certain specific proteins, what are called as transcription factors, will bind, and they make the environment for the binding of RNA polymerase to the promoter. So we can say that these boxes, whether it is pribnow box in case of prokaryotes and Hoagnes box in case of eukaryotes, they have got specific sequences. For example, pribnow box, it has got TAAT sequence, and Hoagnes box, it is also known as TATA box. This serves as the sequence specific binding sites for the transcription factors, then which help us in the binding of RNA polymerase to the promoter. So these are conserved sequences in the promoter of the prokaryotes and eukaryotes. Then we have got another one, what is called as these then we have got another that is called as open reading frame. This is called as open reading frame. What do you mean by open reading frame? This open reading frame, this is called as open reading frame. This open reading frame, it represents the structural genes. It represents the structural gene. This open reading frame, it represents the structural gene. Then we have got another, we are discussing open reading frame. This open, we have discussed the promoter, then we will discuss the another, that is structural gene or open reading frame. This open reading frame, it represents the base sequence. This open reading frame, it represents the base sequence which starts with initiation codons, and ends with termination codons. In DNA sequences, this open reading frame, it starts with initiation codon and ends with termination codon. That means this open reading frame, it contains what? It contains codon triplets which specify amino acids, which encode proteins. That means these are having triplet codons which specify amino acids, For example, if we take this is what is genomic, this is genomic DNA sequence. We have this is genomic DNA sequence. Obviously, in case of DNA, we know that in case of mRNA, we have initiation codons AUG, while as in case of DNA, it would be A, then TG. This ATG, it codes for what? It is for the methionine. That is, it starts, this open reading frame starts with and begins with this initiation codon and ends with the stop codon. Stop codon, we know that we have UAA, UAG, UGA. This is in case of mRNA, but here we are discussing in case of DNA. So in place of uracil there would be thymine. So for example, we have TGA. It is also, TGA is also a stop codon. So this site to this site, this is called as open reading frame. Open reading frame does not contain the stop codon. So we can say that this open reading frame, it can help us. What is the use of this open reading frame? Open reading frame can help us to detect where the actual gene is, where the gene is, where the base sequence which codes for the amino acid is. This is called as gene prediction. It's called as gene prediction. We know that this open reading frame, it begins with the initiation codon and ends with the stop codon. So the base sequences which are present in between the initiation codons and stop codons, they represent the open reading frame. This is open reading frame. So these open reading frame, these are sometimes called as structural genes which encode for the specific proteins. That means they have reading frame, they have codons which specify the amino acids. So this is called as what is called as open reading frame. Then we have got terminator. Then third one is called as terminator. That is called, third one is called as terminator. This terminator, it is the site where site for termination of transcription. It is a site for termination of transcription, and this terminator site, it is rich in guanine cytosine rich region. This terminator region, it is the site which ends, this stops transcription, and this region is guanine cytosine rich region. That means in this region there is the base sequence guanine and cytosine are present in abundance. So this was regarding the what do you mean by transcription, what do you mean by this transcription unit, open reading frame, what do you mean by this promoter, structural gene, and terminator. We also discussed regarding the RNA polymerases. Thanks.